Welcome, everyone, to this lecture on the principles of immunology. Today, we are going to explore one of the most groundbreaking advancements in modern medicine, the applications of monoclonal antibodies. If you have ever wondered how rapid pregnancy tests work, or how doctors can target cancer cells without harming the rest of the body, you are in the right place. We will build our understanding from the ground up, looking at what these molecules are, how they function, and the mathematics behind their binding affinity and clinical dosing. Let us dive in. Before we look at the biology, let us start with a simple analogy. Imagine you are in a massive, crowded stadium, and you need to find one specific person wearing a unique red hat. If you send a regular search party, they might get confused and bring back anyone wearing red, causing chaos. Now, imagine you have a highly trained, laser-focused robotic drone programmed to recognize only that exact shade of red and specific logo. It ignores everyone else and latches directly onto the target. In the human body, monoclonal antibodies act exactly like these drones. They hunt down and bind to one single specific target, ignoring everything else. Now that we have a mental picture, let us define what these are scientifically. Monoclonal antibodies, often abbreviated as MABs, are laboratory-produced molecules. They are engineered to serve as substitute antibodies that can restore, enhance, or mimic your immune system's natural attack on specific cells. The word monoclonal is key here. It means that every single antibody in the batch comes from a single clone of a unique parent immune cell. Scientists typically create these by fusing a specific white blood cell, called a B cell, with a cancer cell, called a myeloma, to form what is known as a hybridoma. This allows the cells to multiply indefinitely while producing the exact same antibody. Understanding how they are made leads us to their most important property: absolute uniformity. Because monoclonal antibodies are exact clones of one another, every single molecule in the batch binds to the exact same part of an antigen. This specific binding site is known as a single epitope. To put this in perspective, natural polyclonal antibodies are a mixed batch that will bind to multiple different parts of an antigen. The absolute uniformity and specificity of monoclonal antibodies mean they do not get distracted. This makes them incredibly powerful and reliable tools in both biological research and medical treatments. This incredible specificity brings us to how they are actually used in the real world. The discovery of monoclonal antibodies has completely revolutionized modern medicine. In diagnostics, without them, we would not have the rapid, at-home pregnancy tests or the rapid antigen tests we rely on to detect viruses like COVID-19. In therapeutics, they are life-saving. They can specifically target and destroy cancer cells without harming surrounding healthy tissue, which is a huge advantage over traditional chemotherapy. They are also used to treat autoimmune diseases like rheumatoid arthritis by binding to inflammatory molecules to stop joint damage. Furthermore, researchers use them constantly in the lab to tag and track specific proteins. Before we move on to the mathematics, let us see this targeting visually. On the screen, you are looking at a diagram that perfectly illustrates monoclonal antibody specificity. Notice the Y-shaped structures? These are the monoclonal antibodies. On the left, we have a cancer cell covered in specific triangular markers, which represent the epitopes. You can see the antibodies fitting perfectly onto these specific epitopes, like a key in a lock. Now, look at the right side. There is a healthy cell with completely different rounded surface markers. The antibody ignores this healthy cell entirely because the shape does not match. This visual demonstrates exactly why these therapies can attack disease without causing widespread collateral damage to the patient. Now that we understand what these antibodies do, we must ask, how do scientists know if a specific antibody is actually effective? The answer lies in measuring how tightly the antibody binds to its target antigen. In immunology, this tightness of binding is called affinity. If an antibody falls off its target easily, it will not make a good drug or diagnostic test. To quantify this, scientists use a mathematical representation called the dissociation constant. Conceptually, the dissociation constant tells us the exact concentration at which half of the antibody binding sites are successfully occupied by the antigen. Let us look at how we calculate this. This brings us to the mathematical formula for the dissociation constant, denoted as K sub d. This formula comes from the principles of chemical equilibrium. The numerator represents the components floating freely in solution, the concentration of the free antibody multiplied by the concentration of the free antigen. The denominator represents the successful pairings, the concentration of the bound antibody-antigen complex. All of these are measured in molarity. Here is the crucial physical interpretation. If the complex forms easily and stays together, the denominator gets larger, making the overall K sub d value decrease. Therefore, a very low K sub d means the antibody has a very high affinity for the antigen. Conversely, a high K sub d means weak binding. Let us see this formula in action with a concrete example. Imagine you are developing a new rapid diagnostic test for a virus. In your lab, you measure the free antibody concentration to be 2 × 10⁻⁶ molar, and the free antigen is 3 × 10⁻⁶ molar. The bound complex is measured at 6 × 10⁻⁴ molar. We substitute these exact values into our equation. First, we multiply the two values in the numerator, giving us 6 × 10⁻12. Then we divide by the denominator. The 6's cancel out, and subtracting the exponents gives us a final answer of 1 × 10⁻⁸ molar. Because this value is very small, it proves the antibody binds tightly and reliably, making it perfect for your diagnostic test. Now that we have calculated how well an antibody binds in the lab, let us shift our focus to the hospital. When monoclonal antibodies are used as targeted therapies for autoimmune diseases or cancers, they are incredibly potent. Because of this potency, they cannot be given as a one-size-fits-all pill. They must be dosed very carefully based on the individual patient's body weight. Accuracy here is a matter of patient safety. If the dose is too low, the drug simply will not work. If the dose is too high, it could suppress the patient's immune system to dangerous levels. Therefore, medical staff rely on a specific clinical calculation. This requirement leads us to the standard clinical dosing formula. D equals W times D. It is a simple but critical equation. Capital D represents the total required dose of the monoclonal antibody that will go into the IV bag, measured in milligrams. Capital W is the patient's total body weight in kilograms. Finally, lowercase d is the prescribed dosage rate, which is established by clinical trials and given in milligrams per kilogram. The physical relationship here is direct and proportional. If a patient's weight increases, the total required dose must also increase proportionally to ensure there are enough antibody molecules in the bloodstream to effectively treat the disease. Let us apply this dosing formula to a real-world medical scenario. Suppose a patient weighing 75 kilograms is prescribed infliximab, a monoclonal antibody used to treat rheumatoid arthritis. The standard required dosage rate for this specific treatment is 5 milligrams of antibody per kilogram of body weight. We need to find the total dose. We substitute 75 for W and 5 for D. Notice how the units align. Multiplying kilograms by milligrams per kilogram means the kilogram units cancel out perfectly, leaving us with just milligrams. 75 times 5 gives us 375. This means the nurse must program the intravenous pump to deliver exactly 375 milligrams of the drug to safely and effectively treat this patient. We have covered a lot of ground today, so let us review the key takeaways. First, we learned that monoclonal antibodies are incredibly precise, laboratory-engineered proteins designed to act like homing missiles, binding to a single, specific epitope. Second, we saw how this absolute specificity makes them indispensable for everything from rapid pregnancy tests to life-saving cancer therapies. Third, we explored the mathematics of their effectiveness, learning that a low dissociation constant, or K sub D, indicates a very high binding affinity. Finally, we saw how this precision translates to the clinic, where dosing must be carefully calculated based on body weight. You now understand both the biology and the math behind one of medicine's most powerful tools.